Hello, my name is Luis Flores, and I will be presenting my poster today. We are interested in determining whether the nuclear morphological changes that occur during oncogenesis, if these have a mechanistic aspect to transformation, and do they contribute to the process of transformation itself. We've known for a long time that normal cells typically display a normal and uniform cell structure along with nuclear architecture and structure. However, upon transformation, we see many changes in the heterochromatin, variations in nuclear size, changes in karyoplasmic ratio, and we've used these features for cancer diagnosis and prognosis. However, as I previously said, how these alterations occur mechanistically and do they contribute to oncogenesis remains unknown. Now, to study this, we use mouse PDAC cells that have a doxycycline-inducible KRAS. Upon addition of doxycycline, we can activate oncogenic KRAS. And we can see here in figure A that there are huge changes from the minus to the plus KRAS. When we activate KRAS, we have a significant decrease in surface area and volume. And here we show that our system in figure C is working well. When we activate KRAS, we see phosphorylation of ERK. Next, we decided to do a screen for nuclear amyloid proteins. We looked at lamin B1, lamin AC, and these three nuclear amyloid proteins. We saw changes in several of these, but we focus on emmerin due to the fact that it is known to be phosphorylated by ERK. For figure F, we knocked down emmerin in these active KRAS expressing cells, and we noted that when we deplete emmerin, we rescued the nuclear size and nuclear surface area and volume of these cells. Next, we decided to investigate some of the more detailed characteristics of these elevated emmerin protein levels. So as we can see here, there's increased emmerin protein. However, in figure B, we know that there is not a significant increase in mRNA synthesis. Next, we postulated that perhaps the increased protein levels could be due to increased stability. Therefore, we performed a cyclohexamide assay, and we noted that there is a difference in their half-life. By the 12-hour mark in the minus KRAS, there's a significant reduction of protein, whereas in the plus KRAS, those levels have not really changed. Next, we wanted to look at more biological aspect of emmerin in these active KRAS cells. We knocked down emmerin using siRNA, and we note by the 48 and 72-hour mark, we have significantly decreased the proliferative capacity of these active KRAS cells. And now we shift more to a nuclear architecture related aspect of our project. Briefly, we want to study LADs, lamina-associated domains. These are domains of the DNA of a gene, of a genomic area that is typically, that will be associated with the nuclear periphery and marked with H3K9 dimethylation. And when we shift the movement of this gene away from the periphery and decrease H3K9 dimethylation, that is typically, those genes are typically increased in transcription activity. So to begin to study this, we do minus and plus KRAS. We do a super resolution of H3K9 dimethylation. We know that there's a significant enrichment when we activate KRAS, a significant enrichment of this histone mark at the periphery as opposed to the minus KRAS. You can see a more diffuse signal here. And to us, that indicates global changes in lamina-associated domains. Next, we perform our Rna-Seq. And I want to skip to C, so it's a more easier figure to understand. So what we do is we plot the gene positions along the x-axis and the fold change along the y. And what we do is we filter out for contiguous genes, so genes that are right next to each other and that are co-regulated in the same fashion, either increase expression or decrease expression. And so we have our blocks selected, and I focus on this example block here. We have these three genes on chromosome 8, and we can see that the blocks are made up here and they're all being repressed. And to us, indicating that these could be possible lamina-associated domains, since there's a large region of the genome that's together and repressed. We are also doing ChIP-seq to integrate all this data and get a definitive answer of which are the LADs that are changing and whether there are key tumor suppressor genes in those LADs. To conclude, we demonstrated that KRAS induction led to significant size reduction of the nuclei in both surface area and volume in these PDAC cells. We saw that when we activate KRAS, we stabilize emmerin. And then when we deplete emmerin, we not only rescue the nuclear size, but we also reduce proliferative capacity. Additionally, we show that there's enrichment of H3K9 dimethylation when we activate oncogenic KRAS. And as I previously mentioned, we are using our RNA-seq analysis and are now processing our ChIP-seq analysis, or ChIP-seq data, to integrate this together and get an idea of what the labs are that are interesting to us and how they are changing. And with that, I'd like to thank Martine for allowing me to work on this project, and the group that I work with for their support and their insight with all the questions that I've had. Thank you.